Good afternoon, everybody. Sorry that I'm here today. We have a tournament in Wairika, so I'm going to be gone, but I wanted to make this video to make sure that everybody understood exactly what to do today. So you should have Miss Smith as a sub today. Be nice to her. But she has direct instructions for what you guys are supposed to do, but I want you guys to take it upon yourself today to really try and get as much work done as you can today, because I'm going to assess basically where we are tomorrow by what you get done today. So you should be done with the quiz. Should be turned in. Make sure that's turned into the box. Should have got 20 minutes to do that. Should have been pretty easy, and once you're done with that, please turn it in. You should have only used your notes, nobody else's notes, so I hope everybody did that, and it should be graded within the next couple days. So now your job is to complete the notes on cellular respiration. So yesterday we got to the point of where glycolysis occurred in the cytoplasm of the cell. It's where it's a breakdown of sugar into a 3-carbon molecule called pyruvate. In this process, 2 ATPs were made. Then after that, you guys took notes on fermentation. There's 2 types, lactic acid and alcoholic fermentation. These both occur when there's not oxygen present. The reason they happen is so that glycolysis can continue. So lactic acid, that occurs in your muscle cells a lot of times, and that's what causes cramps. But that occurs because there's no oxygen present. And when there's no oxygen present, that cycle of glycolysis needs to continue, and without that, our muscles can't continue to make the little amount of ATP, those 2 ATPs that are made during glycolysis. There's also alcoholic fermentation, which happens in yeast. And yeast perform this because most of the time they're in an environment in which there's no oxygen. So that's all they have is glycolysis to make energy. So to continue that on, they do alcoholic fermentation, once again, so that glycolysis can continue and get those 2 ATPs out. So you should be that far in your notes. And once Miss Smith removes the poster board off my whiteboard, there is a completed picture from what we started yesterday. So I think I got through about fermentation yesterday. And then off to the right, you'll see a blue structure, which is the mitochondria. The rest of these 2 processes, which are aerobic respiration, happen in the mitochondria. That's why it's called the powerhouse of the cell. So what your job is today is to complete the notes. On aerobic respiration, that requires oxygen. So you notice on the board that the picture's quite detailed. And what you might want to do first is actually go up and study that picture, draw that picture in as much detail as you can, remembering that I only want you to know the information that explains the picture, that explains the basic chemical reactions. Don't use those big words that you'll find in the textbooks and that you'll find on the internet to describe the Krebs cycle and that describe the electron transport chain. I want you to only describe it to the point of where you know what goes into each process, what comes out, and what the ultimate goal of cellular respiration is. So define it in your own terms. Don't define it, but describe it so that you know the chemical reaction, where it happens, why it happens, and what is produced. So concentrate on that. If you're using any words that you cannot spell or can't even pronounce, those aren't your own words, so don't use those. Okay, so concentrate on those ones that you can use to describe these processes. So as you look at the board, you'll notice that now we have oxygen going in. Now we have that pyruvate molecule going into the mitochondria. And all of that stuff that happens for the ultimate goal of this, making energy. So how much is made, where it's made, that's what you need to concentrate on today. So once you finish these notes, keep these, put them away in your binder, and make sure that you keep them for tomorrow. I'll be here all day tomorrow, so we will talk more about this tomorrow. So if you don't completely get it, don't panic. Okay, I'm going to discuss it. We're going to discuss it as a class. And once again, that discussion is based off what you guys bring to the table. If you guys don't have this done and the discussion stops, then we'll just stop. Okay, and this is up to you. When you're all done, you should have had your hypothesis for your photosynthesis lab written today. If you don't have that written, you need to get that done. Okay, and then tomorrow we're also going to write the introduction. So if you don't have your hypothesis written, get that written. And remember to follow the key steps and make sure you focus on the stuff that it tells you to write. There was a part of that that told you to refer to the equation for photosynthesis. That does not mean write the equation for photosynthesis. That means how is your independent variable affecting that reaction of photosynthesis? Where in the reaction? How is that going to affect that chemical reaction? That's what it's all about. We are not monitoring plant growth. We're monitoring how your independent variable affects the chemical reaction of photosynthesis. So remember that. So then you should move on once you're done with the hypothesis. We're going to grade these tomorrow in class. You should move on to your introduction. Very detailed. It tells you paragraph for paragraph what to write. The first paragraph should be describing photosynthesis. Make sure that all the terms and all the concepts that are covered in your handout are covered in your introduction. Second paragraph, what's happening in your experiment. Once again, make sure that you follow the instructions exactly. What I suggest you do is write a scratch copy first. That means get out a blank piece of paper, write your introduction on a scratch piece of paper. Bring that tomorrow. Don't write anything in the introduction section of your lab handout until we've all looked at what your scratch outline is for an introduction. Do you guys understand that? I hope so. Because tomorrow, this should be blank. The introduction portion of this, you should have on a separate piece of paper your scratch copy, your first draft of what the introduction is going to be. Okay? So I will see everybody tomorrow. I hope this was helpful. And have a great day. Happy Wednesday.